Who don't know, iOptics is our local joint chapter, student chapter of SPIE, IEEE Photonics, and the OSA. We're going to have some pizza for you after, but if I could ask that you guys sign in. If you could write your name right here, and if we already have your email, just like put a dash or something. But otherwise, we do need to keep track of attendance so that we can keep providing you pizza. So please do sign in. I'll start passing this around, and I'll introduce Tan, who will introduce our speaker.
Hi everyone, I guess this is a good time to get started. I'm Tan. I work with Dr. Gabriel Bukascu in the QPI lab. So today it's a great pleasure for me to introduce you Professor Paul Seven from our campus, University of Illinois at Urbana-Champaign. He's from the Department of Physics. So he received his PhD from UC Berkeley in 1990. It was in physics, but actually it was in biophysics. Professor Servin, oh, he got a long CV here with lots of interesting points.
You can cut it down to...
Yeah, I'll just skim through and cut the most important part. So he worked on two trials of fluorescence resonant energy transfer called FRET, based on lanthanide, and another called luminescent resonant energy transfer, or LFRET. And after joining the Department of Physics at UIUC, he turned more interest into molecular models. And then he invented a technique called FIONA, which is short for fluorescent imaging with 1 nanometer accuracy. And that involved a lot to the topic of today's seminar called super-resolution imaging of AMPA receptor as a force structure in live neural synapses. Welcome, Professor Simon.
Thank you very much. One thing, a couple of things. One, I generally go by Paul, so you can call me Paul. The second thing is I'm really looking forward to the pizza at the end. The third thing is, as I'm going on, I'm making various points, and sometimes you'll get them, and sometimes you might be a little confused because maybe I'm not being very coherent. So I'd appreciate it if you ask questions, and that makes things significantly more interactive and I think exciting. So today what I'm going to talk about is super resolution imaging. By super resolution I mean being able to determine not at the micron scale, which is the usual way you image, but being able to image at the nanometer scale. And we're going to particularly apply this to neuronal structures. In fact, as I'll show early on, I did a fair amount of this work associated with molecular motors inside of a cell. You know, it's very busy, and things need to move from point A to point B, and it's the idea of molecular motors that do that. Now I've actually turned to neurons, and to understand how the proteins involved in neurons move around and or are separated. And one thing that we're doing is we can actually look at live neurons, which is fairly unusual, and that usually you take a sample, some neurons, and you fix it. Fixing it is a nice way of saying you kill it, or it's dead. But you can't always do it on live neurons, though in our case we actually can. Okay, so because I assume all of you are not super familiar with neurons, we'll go through a little bit of the biology just so you have a background, and then we'll go into the physics being what the techniques where we get super resolution are, and then we'll come back to the biology and apply it. Okay, so first we want to talk about the nerves, and in particular we're interested in what is called the nerve synapse. in your brain, this is amazing. You have a neuron is the same thing as a nerve cell. You have 80 to 100 billion neurons in your brain. That's an awful lot. One neuron, in general, talks to another neuron. And it does it, so this is one neuron talking to another neuron. It isn't connected to it. There's actually a little space. And what happens is a nerve impulse, some electrical impulse, causes some vesicles to bind to the membrane to release its small molecule in here. Because this is so small, typically only 30 nanometers across, the molecules diffuse and diffuse rapidly, then interact with receptors over here, cause an impulse, and the nerve impulse is propagated from one nerve to another. So a typical nerve in your brain talks to about 1,000 other nerves. So that means if you start out with 100 billion neurons, you're talking about 100 trillion synapses all in your own brain. I mean, it's like mind-blowing. Okay, what we're interested in doing is actually we're interested in studying a single synapse. And where are the proteins? Over here, and this is called the post-synapse. And where are the proteins over here called the pre-synapse? And in particular, what we're interested in doing in the post-synapse, there's something called the AMPA receptor, which is an extremely important receptor because it's how you learn, what memory is involved, and also when you have strokes. For example, I don't know if you notice, but my right leg and right arm is kind of messed up. About 10 years ago, I was hit by a car. I had a brain bleed, and about something like the size of my nail, of my little finger, a certain number of neurons died. And in that, so I have a little bit of difficulty. So it's very clinically worthwhile to understand how these things live, how they die. Or, for example, take a look at my face. Yeah? You remember it? It's good face? Okay. And now, do I have a beard?
Yes.
Okay. So, in fact, so you remembered the fact that I had a beard. How did you remember that? Well, what happened is there were impulses coming from your eye into your brain. Those impulses caused more of the AMPA receptors to congregate in certain synapses, and that's you remembering. So the business of how the AMPA receptors get in particular synapses is very worthwhile to understand. Okay, so how are we going to do this? Well, one thing, we're going to use fluorescence because it's incredibly sensitive. In fact, we'll do single molecule fluorescence. Ordinarily, you do fluorescence in a microscope, you see just 2 dimensions. In fact, you want to see 3 dimensions because in these so-called spines, over here is where this is a postsynapse and there's a presynapse, another neuron coming in, and right here is where we're interested in looking. But the synapses can be anywhere. So we need to do it in 3 dimensions. with nanometer resolution. If we're going to do it with nanometer resolution, we need fairly small probes. We also need them to be photostable because, that your sciatica nerve goes from here down to the edge of your toe. I mean, it can be 1 meter long. So if a nerve receptor or similar protein, it can take a while. In fact, it can take like a month for it to go up here to here. So if you want to be able to see that, you have to be able to look for a month. And so the probes must be also photostable. That is, when you shine light on it, most probes, they emit light and then they die. Typically, they emit for about a second or maybe 10 seconds. We want these to be able to live indefinitely long. Okay, as I said, we want to do it in live cells, and we want to determine the position of the AMPA receptors. Okay? And one thing, so again, the AMPA receptors are right here at the postsynaptic density synapse. And there's also lots of accessory proteins that play a role in forming the synapse, forming where the AMPA receptor are. Okay, is there any questions? Okay, so what techniques are we going to do? First, we're going to look at 3D super accuracy and 3D super resolution. By accuracy, what it means is you can tell how well something is. Resolution is not only accuracy, but in fact, it's separating out two dyes. How close are they? If they're there, it's pretty obvious. If it's here, it can be harder, and even a little closer, it's even harder. So let's first talk about super accuracy, because we want to know where the AMPA receptors are. Okay, and this is what I've become famous for, thanks to Hollywood. So this is Princess Fiona, and in fact, her name is actually an acronym. It stands for Fluorescence Imaging with 1 nanometer accuracy. And in fact, if you take a single fluorophore, what will happen is you typically, it forms an Airy function, which is like this. It can be, you can think of it as a Gaussian function. And if that molecule is just by itself, so it's like this mountain, the mountain may be extremely wide, maybe, a half a mile wide, but the center, if there's enough light hitting your eye, you can determine where it is extremely well. And that's exactly the idea. You just zoom in at an unbelievable level. So a single molecule is right here. It's limited by the diffraction limit to about 250 nanometer wide. But again, if you have enough photons hitting your eye or detector, you can determine where the center is much better than the width. How good can you actually determine it? You can determine the center depending on its signal to noise, and that goes like the width divided by the square root of the number of photons. So that's actually real data. It's on the order of about 10,000 photons. So 10,000 photons, the square root of 10,000 is?
100.
There we go. Got to see if you're still awake. No. For anybody who falls asleep too early, it's no pizza.
That number should be 2.5 nanometers.
Okay, so let's get that. So 250 nanometers divided by 100 is 2.5 nanometers. Now, if actually you do it a little bit more accurately, it comes out to about 1.25 nanometers. And in reality, we could actually determine it to 1.3 nanometers. Okay? So that says if the molecule is here, and then a little bit it's over here, as long as that distance is greater, significantly greater than 1.3 nanometers, we should be able to see it walking. And that's why molecular motors was very attractive to us. The molecular motor is basically this little thing which has 2 feet attached a fluorophore. This is what's called a quantum dot, which is a very stable fluorophore, though we've done it with organic dyes. You have it walk along its roadway. You give it some gas, namely ATP, and there it goes. So you're actually seeing a single kinesin walking. Kinesin is a type of molecular motor. And in fact, you can look at its position as a function of time. So its position is here. It eats a gas molecule. It walks and then waits a little bit, blah, blah, blah. And in fact, we can then plot out the size of this, and we find that it's 8.4 nanometers per step, which is actually in very good agreement with other techniques called optical traps, where they got 8.3 nanometers. Okay, so this basically proves that the technique works. Okay? It works to get super accuracy. Okay, now again, we want to understand super resolution. And here, if you have a single molecule, which we just said, its width is about 250 nanometers. But then if you put another single molecule there, if the distance is much greater than 250 nanometers over here, the point spread function, this guy, is clear, and it's obviously different. If, however, they come on the order of 250 nanometers or less, it becomes impossible to separate them. Okay? Actually, it's not so impossible in that, let's say you have 2 molecules, and let's say one of the molecules disappears. It blinks off. In that case, then you have 1 molecule. You can get it to about 1.5 nanometer accuracy. And then it comes back on, and the other guy blinks off. In this case, you have another 1 molecule. You can get this to 1.5 nanometers, and then you can get the difference to, on the order of 1.5 nanometers times the square root of 2 or something like that. And that's basically the idea. In general, it's called POM, activated localization super resolution microscopy. So you have something like that. You shine normal light on it, and you see basically each of these, because it's about 250 nanometers, you can't see it at all. So what you do is you come in with very light, and you happen to excite this one, this one, this one, so forth. And then you can determine it, each one, to within, like on the order of 1.5 nanometers, because you know where the center is. And then things photobleach. So you have it. And then, in fact, you just go back and you excite with weak light again and repeat it over and over and over again until finally you have it. So that's the technique that has really revolutionized microscopy. because the limit was always like on the order of 250 nanometers, and now you can go down to certainly a factor of 10, sometimes 100. Okay? Are there any questions? Yes?
Is there a limitation of the resolution by this technique?
From what I've said, there is no limit. If you can get enough photons hitting your eye or hitting your detector, you can see infinitely well. In fact, you can see at the angstrom level. The problem is most fluorophores, they put out a certain number of photons, and that's all. And in fact, that's one thing that I'm going to deal with, is that if you can get fluorophores that are really good, you can do much better. Other questions?
So you're not limited to the type of fluorophore. You can basically any fluorophore is a possible candidate. I mean, better to degrees of better or worseness.
That's right. Now you always have a problem that this is assuming that there's no background. And if you have background, then it depends how big the background is. And in general, so you have to have fluorophores that give you at least a signal to background, which is better than 1. Yes?
Well, how do you selectively excite those molecules? I mean, you use a structured light?
No. In fact, you don't. In that you just put in a weak light such that on average, the molecule is excited less than 1 molecule per diffraction limited area. So you don't actually know offhand which one is going to be excited, whether it's this one or this one or this one. That's why you do it multiple times, and eventually you get the whole structure. So in fact, this is also called POM, I don't know, something. It's also called STORM, which is stochastic optical resolution, because it's stochastic how you initially excite them.
So first, they're just randomly excited.
That's right, just randomly exciting.
How many cycles you typically need to
Okay, many thousands, depending on how many fluorophores there are. But one problem is this technique is fairly slow. Typically takes a half hour for you to image it. Now, in fact, as you'll see, we've improved that to like a minute. So you can do pretty well, but that's it. Okay, what I was thinking about is, gee, the rate at which we're going, we'll have to have the pizza and then, come back here. Okay, last thing, we also want to do this in 3 dimensions. Turns out, fortunately, that's actually easy. You put an astigmatic lens in front of your CCD. If you're right in the focal length, it looks normal. If the fluorophore moves down, then what happens is that the image is large this way and small this way, and by taking the ratio, it will give you the Z dimension. And if you go the other way, instead of going like this, it looks wide and then narrow, and you can get the Z resolution also. Okay, so what you do is you label your sample. In this case, it's neurons. And here, this is one dendrite, a nerve cell. And each of these is going, you can see the postsynaptic and the presynaptic density in red and blue. And remember what I said is there should be a distance between them that's about 30 nanometers. Well, here you can actually see 30 nanometers. Here you can just see it, and here you can't. Now this is with regular fluorophores. Not quite, but they're a GFP analog. GFP is green fluorescent protein, so you can transfect them, and they go to the synapse perfectly. The problem is that fluorescent proteins are not very stable. They'll stay lit up for a couple of seconds and then shut off. Okay. Okay. So what other probes? Well, we have fluorescent proteins, as I just mentioned. These are okay. They're small, but they don't last very long. And they can go and label the AMPA receptors right here on the surface, or can in fact label the accessory proteins inside. Or you can use commercial quantum dots. Quantum dots are really fantastic fluorophores in that they last basically forever. The problem is a quantum dot that you buy is about 20 nanometers in diameter, really huge, such that it barely fits in here. So if you're interested in seeing the AMPA diffuse around, the fact that it's a large quantum dot can make it difficult to interpret. On the other hand, what we've developed is small quantum dots, which are about actually 6.3 nanometers. It's in general less than 10 nanometers. They're potentially great because they're fluorescing, they're small enough, and you can see inside of a neuron. Yeah?
If I understand correctly, the big problem with quantum dots is being able to attach them to anything.
Yes, indeed. The quantum dots are actually semiconductors. They're cadmium and selenide. And yeah, they seem fantastic, but you go and you try to label something biological, and the biological thing is really unhappy about it. And you tend to get a lot of non-specific labeling. We have solved that completely.
Completely?
Well,
You could be rich beyond your wildest dreams.
it has crossed my mind. In fact, we've, yes. Yes.
Well, I'd like to talk to you more about that.
And in particular, actually with neurons, it turns out that they're extremely difficult to label because neurons automatically tend to be really sticky. And so if you're trying to label them, that means it's really, really hard. Okay? Okay, so first, we want to do 3 dimensions, and let's talk about the regular commercial QDots. So what we've done in blue, we've genetically transfected. This is a protein, generally called Homer1, but doesn't really matter. Basically tells you where the synapse is. So in this case, these are live neuronal cultures. You have one dendrite here, another dendrite nerve right there. And what we've done is we've taken the AMPA receptor, we've labeled it with our quantum dot. And again, the commercial quantum dots are actually very good in terms of the ability to label and not have a ton of non-specific labeling. The problem that, okay, I won't go into the problems. Okay, and yes?
Just a quick question, I'm sorry. So blue is only a presynaptic stain?
Is a postsynaptic stain.
Okay.
And this is, then red is the AMPA receptor, okay? And so what we do, again, remember we do it in 3 dimensions. So there's the AMPA receptor in 3 dimensions. This is done with Klaus Schulten's VMD. It's really quite amazing. Okay, so you see there, and then actually this is a single AMPA receptor diffusing around with a time resolution of about 100 milliseconds. And you see eventually you get this.
This is within the cytoplasm.
Oh, this is on the extracellular side because what?
Sorry, is it the AMPA receptor or the transmitter that you are?
Okay, hold on. So you have, here's one nerve, here's another nerve. The axon from this nerve comes in, it interacts with what is called the dendrite of this. And notice the AMPA receptors are on the surface. So in fact, you come in, you label them right here. You do not need to go through the membrane and label anything over here with the AMPA receptor. On the other hand, we've genetically labeled things that are inside right over here, so we know where things are. Is that clear?
Yeah.
Maybe.
Well, what you're releasing is the neurotransmitter, so that's what's going to be in the extracellular space.
Yes, that we are releasing this into the extracellular space. Now, in fact, how much of it we don't know, and at this point we don't actually care. We're just looking at sort of the steady state level. At some point, it's called LTP, long-term potentiation, we actually will look at it, and hopefully I'll get time to talk about it. Okay, so once again, we see with big QDots, it seems to work. And in fact, sometimes it does work. And I'll show you in a minute, other times it doesn't work. Because it turns out that what happens is sometimes this synapse is big enough that the QDot can go through, and other times it can't. Okay, so this, in fact, is a more typical diagram. Here you see, here's a synapse and there's another synapse, and all over here is AMPA receptors with the big QDot. And in general, you see a couple of them are in here, but mostly they're not. Okay? So I'm going to skip this. So what we wanted to do is we wanted to use small QDots, which is basically just the QDot. This is the QDot core, which is about 2 to 3 or 4 nanometers in size. You need a ligand shell because the QDot core is quenched completely by water. So you put a ligand shell, and normally this ligand shell is like about 10 nanometers over here to prevent all the non-specific labeling. In fact, what we've done is we've put a much shorter ligand on here and capped the ligand with polyethylene glycol, which is very water soluble. And lastly, we put something that's a little bit reactive. And in fact, you can see over here, you see this is a neuron where it's a little thicker, indicates there's a synapse there, and notice you get essentially no non-specific labeling. And all of the labels, that is, it goes and finds the AMPA receptor, goes and it binds. And it's unbelievably photostable. Okay, and in fact, here we have the green and the red, which is the AMPA receptor, and yellow where there's overlap, and you can see there's really tremendous overlap. Not only that, but we do the same experiment again. And this was actually a surprise to us. See, we take a look. In blue, we have the postsynaptic density, and the red is the AMPA receptor. We do it in 3D over here. And now notice what happens. The AMPA receptor tends to be in one part of the synapse. Okay, just 1 second. And it turns out that that is probably very important. And what it turns out that if you look at a neuron, typically, although I don't have good statistics on this yet, it's typically the AMPA receptor that at least can be labeled and where it can diffuse is generally less than in 100 nanometers by 100 nanometers. And this actually agrees with some other high-resolution studies that have been done. What the implications for this is really unclear. But for example, in cases of autism, there is definitely situations where it affects the spatial organization of the AMPA receptor. So it might have true ramifications. Okay, you had a question before?
So if these don't, if they're photostable, they don't blink, right? So are you saying that you're imaging just one at a time?
In fact, there's no reason in these experiments for it to blink. It turns out, actually, sometimes it does blink. So actually, we can do high resolution. But in this case, because we're looking at dynamics, all that we really care is that it's basically on. It's on, and it's on, it's on, and on. That's all we care about. We don't actually ever have to have them blink. It turns out they do also, but that's sort of another story.
But your labeling is specific enough that you're only getting like one AMPA receptor in a synapse, or only one exists?
Oh, no. There are probably 100 or 150 of them exist. No, we're labeling very lightly. Okay. Furthermore, we can tell the difference between the small QDots and the big QDots. The small QDots are basically, they go inside the synapse and are basically stuck. so they have a low diffusion constant. On the other hand, when we do big QDots, we see actually two populations. Over here is like the free diffusion due to the AMPA receptor not being in the synapse. And sometimes we see over here, which actually agrees very well with this, that is sometimes the big QDots do get into the synapse. And it's essentially stuck inside of this. Okay? Okay. And then 12:44. So let's see, so we should probably go another 5 minutes or so.
Do 10 probably.
Okay? It's really dangerous to, you think you're being nice, but hey, you know, we could talk, I could talk all afternoon. Okay, so now actually we're going to come to the second part of my talk. So let's see, so that's probably another 45 minutes. That's okay. And that is using the fluorescent proteins to label them. And the big advantage of the fluorescent, well, let me say first, the disadvantage is they don't last very long. Though actually we find that after a lot of searching, they last long enough that we can get 10 or 20 nanometer resolution. So you're not doing too badly. The advantage of them is you can label whatever you want, and in particular, you can label intracellular proteins, either in the postsynapse or in the presynapse. And I'll show you both of them. And in particular, in the postsynapse, there's two proteins that we happen to be most interested. One is called postsynaptic density 95, and the other one is called HOMR. Shank, actually, we haven't done, but this is probably of interest too. And what we're interested in doing is doing dual color labeling. That is, looking at the position of the PSD 95 and the HOMR. Or we're looking at the PSD to something like synapsin, which is a protein in the presynaptic density. So we labeled it with green and with red and so forth. Okay, and first what we do is we do a measurement, for example, from PSD to synapsin, and we find that it's 243 nanometers. Another we do from HOMR to PSD, so that's 331 nanometers. Okay. Then what we're interested in doing is what we call fast palm. That is, we can get this kind of resolution in the order of 1 or 2 minutes instead of the normal 30 minutes. And what you have is, this is time, and this is the PSD. It maybe slightly changes, and the HOMR also significantly changes. And you can, and that's one thing, and there it's growing larger. Okay. And what you do is you look at the distance, in this case between synapsin and PSD 95, and in fact, in general, with time, that is with LTP, it decreases. So in fact, what you have is it decreases in the order of 30 minutes to about 125 nanometers. And in fact, we do the same sort of experiment from PSD to HOMR, initially it was 330 nanometers. Then when you do LTP, essentially chemically you're inducing a memory in the synapse. It goes to 92 nanometers. Okay. The situation is actually fairly complicated, as you find biology is completely different than physics. You know, physics, you learn or you remember certain laws, you know, and usually there's not too many of them. You know, Newton came up with 3 laws, and then you figure out how they apply in different situations. Biology is a fucking mess. Oh, wait, oh, that was recorded. So, as I was saying, biology is a mess. Because in one situation you have one result, another situation you have another result. And for example, here you have a distance. This is in different synapses. Otherwise, it seems that it's the same preparation. In some cases, with time, the synapse is decreasing and then increasing, or it stays the same, or it just increases, or it just decreases. And of course, you know, when we put on our biology hat, we have to try to figure out what's going on. But this sort of information is totally new, in that what people thought in general is that you'd always have a decrease, but in fact, on the same nerve, some synapses you have an increase, and others we see that we have a decrease, and all sorts of things. Okay, so in summary, we've combined 3D super resolution imaging of synapses with receptor tracking, the AMPA receptors, and we can demonstrate that AMPA diffuses in the synapses. We've developed a small quantum dot for labeling AMPA receptors. The small quantum dots are more accessible to the synaptic cleft, and they reveal more details about AMPA diffusion. The AMPA diffusion, that's spelled wrong, is restricted in general to less than something like 100 nanometers in size, where the whole synapse is on the order of 500 to 1 micron in size. And we can also see long-term potentiation associated with the various support proteins with a minute time resolution and on the order of 10 nanometer accuracy. And lastly, I want to thank the people who actually do the work. You know, the good thing about being a professor, you know, we were talking about beforehand, oh, you know, it's a real pain in the butt. Pain in the tushy. Because, you know, you have to constantly go out and ask for money and so forth. But the great thing is, you don't actually do the work. And you have postdocs and graduate students who do the work, hand me the result, and then I get to tell you all about it. So there are 3 people who are absolutely instrumental in this. Pinghua Go. Who's Chinese in here?
Go.
Oh yeah, what is it?
Go.
Yeah, that's what they say. See, what I do is I say, oh, what is that, go? No, he says,
Go.
and then I say, oh, you mean go? And then he says, and then, hold on, and then the third time, I say, this is Pinghua go, and then he says,
Yeah, right.
exactly, exactly. There's something about it, there's 3 times, and basically they give up. Anyways, he's a synthetic chemist that was responsible for making these small quantum dots. And there was a tremendous number of highs and lows associated with it. It's been incredibly difficult, but he succeeded. And this is trouble because her husband is sitting right here. So, Kai?
That's okay.
And whatever has been responsible for all of the AMPA receptor work actually done on neurons. And Sang-Hak Lee, who I think I'm pronouncing that right, he's a postdoc who's been responsible for all of the work, again, with neurons, and this time using the fluorescent proteins. And that's my dog. Lucky. Thank you very much.
So we've got 5 minutes left for questions.
So we have to wait the 5 minutes before we have the pizza?
I guess 1 or 2 questions.
Come on, you got to ask them so then we can...
Do you mind going back to the one where you had the different, yeah, keep going, a couple more. That one right there.
Hmm?
Okay, sorry, I was just looking at the axes. So this is the intensity, but and previously you were looking at the distance between two proteins.
That's right. The intensity also changes because in general, for example, synapsin or PSD95, you find that more of it when you induce LTP, you get more of it. And what we're showing in distance is actually not only are you getting more of it, but it's shifting one way or the other.
Okay. Yeah, but it looked like the spines or the boutons were kind of getting stubbier and closer together.
The boutons or spines definitely change in size. I wouldn't, in general, on average, I would say, yes, it gets stubbier in size. Now that we can analyze, one synapse and another synapse, whether you have, something like this or like that, we'll see what the statistics finally says.
Okay, yeah, because I was just wondering if it had something to do with just the angle of the incident focal volume on the...
No, absolutely not, because this is in 3 dimensions. That's an important point. Yes?
Just a simple question. What is the quantum yield of your small quantum dot in a solution?
We don't actually know, but we can tell the intensity compared to like a big quantum dot. And it's about 1/3 as bright as the big quantum dots. So I don't know if that's good or bad, but the answer is it's plenty bright enough to see a single molecule with very reasonable time resolution. In fact, in this case, you can get down to probably on the order of 10 milliseconds. So it's not quite as good as the commercial ones, but so I figure I'd make, $1 million instead of $2 million off of this. So,
Any more questions? So let's thank Dr. Paul Seven again. And